everyone, good day. Today we're going to talk about case study, which is leader gene danger from unplanned fish breeding, which is the cause of STA6063 advanced genetic from the group of Chin and Wilson. Today we're going to talk about, we have three different parts. The first one, we're going to talk have some introduction, which is regarding genetic deterioration, and then as well as current aquaculture practice. The second one, we're going to talk about what the inbreeding going to bring the threats for aquaculture, and as well as the consequences from genetic deterioration and unplanned breeding. Third one, we're going to talk about suggestion, which is some steps to overcome genetic deterioration and unplanned breeding. The first one for introduction is problem statement. Our problem statement is unplanned fish breeding may cause genetic erosion in aquaculture production and open water fishery resources. This is from Bangladesh Fishery Research Institute 2019. Second, we're going to talk about inbreeding in aquaculture, the mating, which is the mating of animal, which is closely related than the average relationship within the breed or the population concerned. The third one is the effect of inbreeding in fish, because inbreeding gonna decrease the production phenotypes such as growth rate, fecundity, and survival rate. Meanwhile, you're gonna increase the number of deformed offspring as well. This is from FAO. The fourth, we're gonna talk about the effect of inbreeding in human. This is gonna cause genetic disorder due to inbreeding, such as blindness, hearing loss, neonatal diabetes, limb malformation, disorder of sex development, as schizophrenia, and several other. This is from Woodley 2009. The fifth, we're gonna talk about recessive gene disorder. They receive one copy of one gene from each other parent. Animals that are closely related are more likely to carry a copy of same recessive gene. This is gonna increase the risk, and they will pass a copy of the gene into their offspring. This is citation for LIC 2022. The last one, we're gonna talk about genetic deterioration, which inbreeding gonna lead to expression of deleterious recessive allele. In small population, the choice of the mates also limited. Over the time, individual will all become related and will be forced to mate with relatives.
Okay, so for the trend in aquaculture, so I would like to take one case study, which is a paper from India done by Chattopadhyay and Gorai in 2018, which they went to these three popular locations in India for hatchery, for fish hatchery, which is at West Bengal, Bihar, and Assam. So the objective is to assess the current practices correspond with the principal objective of the technology. So what finding is at most 98% of them started with their own technique, and then due to improvement of the technology, they adopted at first the Hapa breeding, after that the Chinese style hatchery, and then when many people knows that doing hatchery has this huge profit margin, which in 4 to 6 months they can get quite a lot of money. Many people start to go into this industry, this hatchery production, and then due to faulty and after a few years of operation, this has caused farmers to do mixed spawning, small breed population, and encourage breeders, and the use of the indiscriminate hybridization means they don't really choose proper fishes for breeding. So the consequences from what the reviewers have said is that 37.6% has deformities, 15.6% has decrease in FCR, 19% has decrease in fry survival, and other points mentioned by the writers were the introduction of alien fishes and the use of unauthorized drugs and feeds has really severely affected the native biodiversity. So the general trends or consequence of inbreeding, actually inbreeding does change gene frequency, but they change the genotype frequency, and it increases homozygosity and decreases heterozygosity. It repeated inbreeding may discard some valuable alleles, and inbreeding causes inbreeding depression. For example, abnormalities in structures and unsize, different in sizes, those kind of things, which was mentioned earlier. Lower egg production capacity, decreased fertility rate. Fry become either large or small, medium-sized fry is not observed. At least 25% of fry die in breeding. Lower growth, increase in expression of recessive genes, means the fish are getting not really healthy every time they breed. And then lower viability, loss of genetic variation within the inbreed population.
This is actually a study being done by this Kin said back in 1976. So the title is actually talking about the inbreeding for rainbow trout in a population, and the result of the study is showing that saying that it's not really applying to all the fish population, because this is basically done on rainbow trout only. But this major finding of these studies basically focus on the production phenotype. As we can see on the graph, majority of the growth production, or you can see the weight, as well as the egg mass weight, as well as the survival rate, all major have a negative impact caused by the inbreeding. And it has been suggested that breeding program used by most fish farmers will produce inbreeding of 3 to 5% per generation. If this occurs, inbreeding depression can begin to affect the productivity and profit after 3 to 5 generations. Next, we're going to talk about suggestion. So one of the suggestion is introduction of unrelated fish to outcross on the existing broodstock. For example, every year you have 10 or 20 unrelated fish should be introduced into the hatchery. As a result, outbreeding will be occur and increase the genetic variation. And unrelated fish must be free from any diseases, so-called SPF free. Yeah.
Okay, another view from our group is that the government side should produce good strains of breeders and provide to local hatcheries, which means they can actually establish what was now quite popular to be used, the term as BMC or NBC, Broodstock Management Center or Nucleus Breeding Center, which allows the good genes and healthy fishes to be produced in the market, and this will certainly reduce inbreeding. Another one is that we do the selection within cohorts and exchange of breeders. So the broodstock could be arbitrarily divided into several groups. Mating could then be performed between individuals from the different groups on a rational basis. to avoid inbreeding. This is also supported by ECNAC in 1991. So as a conclusion, we have 4 points that we wish to share. First is the genetically improved strains are actually essential for aquaculture development. However, consideration must be given to the potential effects of the fish escapees and the conservation of the wild fish genetic resources.
The genetic management is necessary for both genetic improvement and conservation of genetic variation in culture fish population, and should be emphasized in future capacity development initiative. The next one, also the last one, the documentation on culture and wild fish genetic resources will be increasingly important for conservation strategy as new strains are developed and aquaculture production can further expand. This is some of our reference that we get from for our information. Lastly, we thank you for your attention. Thank you.
Thank you.